Good day, everyone. My name is Wei Lun, and I'll be moderating concurrent session 8, entitled Genetic Variation of Raptors. For this session, we have 5 papers. Before we start, for a gentle reminder, each presenter is given 20 minutes max for their presentation, including about 15 minutes of presentation plus a 5-minute question and answer. Okay, these are for live presentations. So for pre-recorded presentations, they may stretch up to a maximum of 20 minutes. Those who would like to ask questions may do so either by unmuting yourself at the end of the presentation or typing your question in the chat room, and I'll try to read them out after the presentation finishes if we still have time. All right. So without further ado, we have Natalia Andreyonkova as the first presenter with the title Genetic Relationships in Black Kites from Northeast Asia, Japan, Taiwan, India, and Australia. Natalia, the floor is yours whenever you're ready.
Thank you, I'm ready, but it must be a pre-recorded presentation.
All right. Could you play it?
But could you play it? As I know, it must be your shot. It must be pre-recorded presentation with sound.
Do we have the tech support over here to assist?
Okay, I'm so sorry. I forgot to share this one. I'll play back the recording.
All right, sure. Thank you.
Dear colleagues, I'll talk about genetic relationships in black kites from Northeast Asia, Japan, Taiwan, India, and Australia. The black kite is one of the most widespread birds of prey. It inhabits most of Eurasia, Africa, and Australasia. The species displays a unique ecological flexibility, and it is found from northern taiga to steppe. The black kite is an opportunistic predator and scavenger. This species is very successful in many parts of its range. It quickly adapts to anthropogenic pressure and may even benefit from human presence, but in some areas, human activity has really hurt it. The black kite is thought to have from 5 to 7 subspecies that occupy vast geographic ranges. The Palearctic is populated by 2 subspecies, the European black kite migrans in the west and the black-eared kite lineatus in the east, including Japan. India and Indochina are inhabited by the pariah kite Govinda, and Australia and its adjacent islands are inhabited by the fork-tailed kite Aphanis. We recently published a study where we showed that populations from Europe, Northern Asia, and India all had separate haplogroups of the mitochondrial cytochrome B gene. The European green group and North Asian red group were isolated in the Pleistocene and then spread across the northern Palearctic following climate amelioration. They have formed a broad intergradation zone from western Siberia and Kazakhstan to eastern Europe. The birds in this zone exhibit the whole range of intermediate external characteristics, and we found that there is a mixture of migrans and lineatus haplotypes here. Moreover, the lineatus, these red haplotypes, has already penetrated to Central Europe, which means that the intergradation zone is expanding westward. However, low variability of the cytochrome B gene didn't allow us to find differences between Indian and Australian populations. They share the same, this light blue haplogroup. So we analyzed in addition another mitochondrial gene ND2, and this allowed us to genetically separate the kites of India and Australia. It turns out that the Australian haplogroup was more ancestral. During the last glaciation, 19,000 years ago, ocean level was so decreased that part of the continental shelf were exposed as land, which is called Sundaland. Black kite could inhabit the whole this area, from India, Indochina and the Malay Peninsula to Australia, with permanent gene exchange between Indochina and Australia. Then the glacier began to melt. Rainfall and humidity began to rise in the Asian monsoon region. This caused ocean level to rise and forests spread across all this territory. The black kite doesn't like continuous forests, since it cannot compete with some more adapted predators, for example with hawk eagles. We assume that it was the forest expansion that led the kite's range to shrink. And it seems that there were two main populations left in Australia and somewhere in the Indus River Basin and in the Thar Desert region, since there remained fairly dry savannas in that time. About 5,000 years ago, monsoon began to weaken again and savannas began to reappear. In India, the Deccan Plateau became drier and black kite population started spreading eastward. Australian kites also started expansion, penetrating the islands. These are the modern areas of Govinda and Affinis. Apparently they continue to spread now. In our genetic research we used samples of Govinda only from India. Unfortunately we have no samples from Indochina, but this area is very interesting. Some enclaves of the ancient ancestor kite population that once occupied all the Sundaland could have remained somewhere here. The territory of Thailand is particularly interesting because savannas remained in the foothills even in the wettest period. It would be great to get samples of the black kite from the northern part of Thailand and from neighboring countries and study their genetics. Maybe somebody of the symposium participants would like to take part in this study. Another interesting unexplored territory is located in southwest of Asia. Kite phenotypes and our genetic data confirm that European migrants, Eastern Lineatus and Indo-Malay Govinda, all they are mixed on this territory of Pakistan and Afghanistan. Most of Pakistan is inhabited by the sedentary Govinda, and Govinda occupies the southern foot of the Himalayas. But the northwestern border of its range remains unclear. In the central Kazakhstan, we found unexpected, unusual haplotype. It belonged to Govinda group, but it was slightly different from all Indian haplotypes. It's possible that this Govinda haplotype penetrated into Kazakhstan from somewhere south. It's quite possible that isolated Govinda populations exist in the Pamirs and possibly in the mountains of Afghanistan and Iran. So the unusual haplotype found in Kazakhstan could originate from one of these unknown Govinda populations. It would be great to get black kite samples from this territory. We could check their genetic group. Perhaps we would have discovered some new unknown kite genetic lines. As I said, we have sequenced 2 mitochondrial genes and identified these haplogroups. Now let's look at this group. It belongs to the kites of Northeast Asia, which are Lineatus. Also, these haplogroups were found in Japan and Taiwan as well. Japan is inhabited by Lineatus, but the Japanese black kite population is believed to be isolated, as ring recovery data suggests only intra-Japan migrations. These migrations are relatively long. It looks like Japanese kite population should genetically differ from the population of the mainland. We examined 19 samples from the island of Hokkaido in Japan. They turned out to be very homogeneous and all belong to one of the two Lineatus haplogroups, this light red group. Unfortunately, mitochondrial genes didn't allow us to learn more. We hope to get more material from other islands of Japan and from Russia and Far East. Maybe one of Japanese colleagues would like to participate in this study. We would like to analyze a number of nuclear genes to determine how long the Japanese population has been isolated from the mainland and how strict this isolation was, and whether there is gene flow between the mainland and Japan. And let's see here now. On the island of Taiwan in 1920, its own subspecies of black kite, Formazanus, was described. It was described as very similar to Lineatus but smaller and with slightly different proportions. Then kites from Hainan and the south of mainland China were considered as Formazanus as well, although there was no evidence that these populations were related to each other. Anyway, much has changed since then. Due to the active use of poisons in agriculture, the black kite population declined dangerously 40-50 years ago. In the 90s, the use of poisons was restricted, and the black kite began to recover in Taiwan. At the same time, Govinda is moving from India, and Lineatus is spreading from the north. So we have no idea who actually lives in this area now. We examined 27 kites from Taiwan. We analyzed these two genes and found both red Lineatus haplogroups in Taiwan. But we also found a third group, a new one that we hadn't ever found before. Here this group is. It clearly, not very much, but clearly differs from all Lineatus groups. It's possible that this group is inherited from the very Formazanus described in 1920s by Kuroda. Or maybe this could be just another group of Lineatus, which came, for example, from the mainland China, from where we have no genetic data. In any case, currently there is a mixed population in Taiwan, so we don't know which of the true Formazanus genes are preserved here. Mitochondria are good because they are passed on through the maternal line, unchanged, without dilution. But nuclear genes, including genes responsible for the phenotype, are diluted with each hybridization. I think we were lucky finding this haplogroup survived after massive extinction of the black kites in Taiwan. Another interesting detail about Taiwan. There are currently two kite populations, the northern and the southern, and these populations appear to be pretty isolated from each other. Here is the data of the iNaturalist website. The red squares are the observation of the black kite. We found that these two populations differ genetically. In the north, we found both Lineatus groups of Northern Asia, green and blue groups. And in the south, this is only there is only the blue Lineatus group and this new Taiwanese red haplogroup. The dots mark the nesting specimens, so we can see that all three groups breed in Taiwan. The climate and kites breeding periods in the south and north of Taiwan are not very different. Kite phenotype differences were not reported as well, so we shouldn't think that more or less pure Formazanus still live separately, preferring the south of Taiwan. No, it seems that kites from the mainland entered Taiwan many times and started to breed here and hybridized with local kites and diluted local genes. In order to reconstruct Formazanus population history, we are going to investigate a number of nuclear genes. However, we need samples from the mainland breeding population. There is very little published about it. It would be very interesting to find out whether there is this new Taiwanese haplogroup in the mainland, or maybe there is something special and new here, and how many Govinda or Lineatus genes are there. And of course, the Hainan population is very interesting because it was also classified as Formazanus many years ago. But still no genetic data is available about all this territory. So we need samples from here too. I think that's all. Thank you very much for your attention.
Thank you, Natalia, for the interesting presentation. All right, so now I'll open, since we have a few more minutes, I'll open the floor for questions. If you have any questions, you can unmute yourself to read out your question, or you can just type in the chat box and I'll try to read it out for you. Any questions? While we are waiting, maybe I can ask a question. From your map over there on the distribution and also the haplotype groups, I see quite a lot of admixture in terms of the subspecies and all from different regions. So from your research, could it be possible that some of the subspecies currently recognized are actually hybrids of two other wider ranging subspecies? What do you think, Natalia?
Thank you for the question. Yes, we found some hybridization zones in Siberia. Lineatus and migrants are hybridized, and in Pakistan there are 3 hybridized subspecies: Govinda, migrants, and Lineatus. And so, as I said, we don't know something about Formazanus, and we would like to learn out something, but we need some samples of tissue, blood, or pin feathers from these local populations, and it would be great if someone of participants would like to cooperate with us.
All right, great, thank you. We have a comment here in the chat box by Tulsi. So in Nepal, we get both Govinda as well as Lineatus subspecies. Lineatus are migrants, so there could be chance for hybridization. It would be interesting to include some samples from this part. So probably you both can connect and share some samples and
yes,
insights.
Sure,
yeah,
sure, yeah. It would be great, yes.
Mm-hmm. You also mentioned that you will be using some nuclear markers to do further structuring analysis. So are you going to use coding genes or other kinds of markers as well?
I'm going to use some coding genes, but I mean not coding fragments of the genes.
Mm-hmm.
I mean, because it's can evolve more faster than coding fragments. So yes, it must be quite fast evaluated genes.
Yep, definitely. Maybe you can look out at some multi-locus markers as well, such as microsatellites. Maybe they contain more information as well.
Yes, of course. I don't know about microsatellites. I'll use it, but I think that microsatellites, it's not very good for evolutionary study, because it's too fast evolving and can evolve in different as there and here, and we can't understand the evolution. So I prefer to use genes, coding genes.
All right, thank you very much for the sharing, Natalia. So since there are no further questions, we'll probably move on to the next candidate. So thank you very much, Natalia, for your time and your sharing.
Thank you.
Next up, we'll move on to the second presenter, Nagai Kazuya, with the title Analysis of Genetic Structure and Genetic Diversity in Japanese Black Kite Population Using Mitochondrial DNA. I understand that the presenter himself is not here, so we'll be playing a recorded presentation. If you have any questions, you can feel free to actually contact the presenter later on. Please go on with the presentation. Thank you.
Okay. Hello everyone. This is Kazuya Naga from Iwate University in Japan. Today's presentation is about black kite. The title is Analysis of Genetic Structure and Genetic Diversity in Japanese black kite population using mitochondrial DNA. So let's start. First of all, I would like to introduce myself. My major is molecular biology, molecular ecology, and population genetics. My main research interest is birds. In particular, my main focus is on the genetics of raptors living in Japan. I have studied and published population genetics of eastern marsh harrier, gray-faced buzzard, eastern buzzard, Ogasawara buzzard, Japanese sparrowhawks, and osprey. I have also conducted and published a study on the Oriental honey buzzard, bacterial flora of hindgut using next-generation sequencing. One of my recent publications that you may find interesting is my research on eastern buzzard. I did a phylogenetic analysis of the eastern buzzard in Japan and the eastern buzzard in Eurasia. This is a part of the figure in the paper published in the Journal of Raptor Research. I reported that the eastern buzzard in Japan and the eastern buzzard in Russia may be different species. The gray area in the phylogenetic tree is the Japanese eastern buzzard, which is a different group from the Russian eastern buzzard. I reported that the Russian eastern buzzard may be Himalayan buzzard because the sequence of the Russian eastern buzzard is identical to that of the Himalayan buzzard. The IOC has now subdivided the eastern buzzard of eastern Eurasia as a subspecies of Japonicus. I think this is a mistake. I think they are different species. The study of eastern buzzard in East Asia is one of my main subjects. Back to the topic at hand. The birds of prey in Japan, black kites, have yet to be studied. I chose the black kite. I am currently doing research on it. This is my report today. Next, I would like to introduce a little bit about black kite. Black kite is subdivided into 5 subspecies according to 2021 IOC bug list. In East Asia, Lineatus, Govinda and Formosanus are found, and Lineatus inhabits north of latitude 30 degrees north. Therefore, the black kite inhabiting Northeast Asia, including Japan, is Lineatus. These photos were taken in Singapore, a country next to Malaysia. I wonder if this black kite is Inukai Govinda. These are the black kite of Thailand. It is displayed as Lineatus, but I think it's Govinda. These are Formosanus Taiwan kite. I think the white of the feathers and wings are wider and darker than the other subspecies. Okay, I would like to introduce the Japanese black kite. It is one of the most familiar raptors in Japan. They can be found here and there. It sometimes flies over my house. They can be found from Kagoshima, the southern tip of Kyushu, to Hokkaido, the northern tip of Japan. It is said to be a legend bird, but its population increases in the winter. It is a short migratory bird and is very well adapted to living with humans. They compete with crows for food scraps produced by humans. They often take and eat human food such as hamburgers. To be honest, they are not very well liked birds because they scavenge garbage and take people's lunch. According to recent observation records, the population may have leveled off or declined slightly when comparing the 5 years from 1997 to 22 and the 5 years from 2016 to 2021. The black kite used to eat human garbage, but due to proper waste disposal, they are no longer scavenging for garbage, which may have reduced the number of cases observed. Therefore, we aim to investigate the genetic diversity and breeding dynamics of the Japanese black kite population. Their populations that should be protected. Is genetic diversity maintained? Is there interbreeding between local populations? How did the Japanese kite come to Japan? Specifically, our goal was to investigate these questions. Using mitochondrial DNA as marker, we conducted a population genetic analysis of the black kite population in Japan. Mitochondrial DNA has the advantage that it evolved faster than the nuclear genomes, and that DNA can be extracted even from slightly degraded tissues. We collect a total of 59 individual samples from 22 sites around Japan. Most of them are feather samples. There are also some fish samples. There are many black-cats in Iwate Prefecture. Iwate is here. Iwate is here. So we have many samples in Iwate. The distance from Kagoshima is here. Kagoshima to Abashiri in Hokkaido is here. Is about 1,800 kilometers. We were able to get samples from almost all over Japan. Method: DNA is extracted from feathers, shafts, and fish samples. Using the DNA as a template, fragments of mitochondrial DNA are amplified by PCR. After the PCR products are purified, direct sequencing is performed to determine the nucleotide sequence. Based on the obtained data, I conducted population genetic analysis such as phylogenetic analysis, network analysis, neutral tests, and mismatch distribution tests. Based on these results, I examined the natural population history and current status of the black-cat population in Japan. Here are the results. We determined 1585 base pair of mitochondrial DNA from 59 black-cat individuals. The number of haplotypes obtained was 6, and 7 mutations were identified. The genetic diversity was 0.304 plus or minus 0.075, and the base nucleotide diversity pi was 0.00044. The obtained haplotypes are shown in the table where in Japan they originated. 85% of the total contained MM1. MM1 was detected from all over Japan. 85% of individuals are genetically identical. The next common was MM5, 8% of the total, detected from almost over Japan. Some haplotypes were regionally specific. Network analysis with 15L5 base pair showed a star shape centered on MM1. MM1 and MM5 differ by 3 base pairs. MM1 and other haplotypes differ by 1 base pair. Okay, perhaps the ancestral haplotype of the Japanese black-cat was MM1, and after MM1 spread throughout Japan, other haplotypes were developed. If other haplotypes are also Japanese ancestral species, more individuals should be detected. Since MM5 and MM1 have a difference of 3 base pairs, they are derived from different populations. Using a barcode cyclone B, we performed a phylogenetic analysis using the data in the NCBI database. The tree shows clusters among the subspecies roughly. However, detailed regional identification is not possible. Milbus is the outgroup, which is divided into the migrants group and the group of migrants, lineators, and Govinda. The group of lineators and the group of Govinda and affinities, respectively. The data obtained in this study are enclosed in red square. The Japanese black kite was shown to be in the lineators group. It also has the exact same sequence as the Korean black kite. Here is a phylogenetically created using parallel control region and the data in the database. The red squares are our data. 5 haplotypes except MM5 are clustered in Govinda. These Govinda are from India and Pakistan, while MM5 is clustered in lineators, which is from India. Since lineators is known to winter in India, it is possible that these Govinda are also lineators. Here is a summary of the results so far. Phylogenetic analysis shows that the site B phylogenetic tree indicated that the Japanese kite was lineators, while the control region phylogenetic tree indicated that part of the Japanese kite was lineators and part was Govinda. In addition, the sequence of MM1 was exactly the same as that of Korean kite. The network results showed that MM1 and MM5 differed by 3 base pairs. Based on these results, the Japanese kite can be divided into 2 groups, the MM1 group and the MM5. We also conducted tests to determine if the population is expanding or maintains stability. I think the Japanese black kite population has been shown to be a population expansion model. Next, this is the genetic diversity of Japanese raptors. Most of my reports, the genetic diversity of Japanese kite is shown to be quite low as that of the Ogasawara buzzard. It is even lower than that of the Eurasian collared dove, although it is not a raptor which has about 100 individuals in Japan. Why is the genetic diversity of the Japanese kite population so low? It is not that the number of individuals is small, nor that the number of individuals has decreased in the past. The black kite is considered to be a relatively new species, probably because a small number of individuals have just recently arrived in Japan. Here is a possible hypothesis of black kite invasion to Japan. The black kite arrived in East Asia. Eventually, individuals with MM1 haplotype will probably invade Japan via the Korean peninsula. This is because the Korean kite and the Japanese kite have the same genotype. The kite that came to Japan increased its population and dispersed throughout Japan. Next, an individual with the MM5 haplotype invaded Japan. Since the sequence was the same as that of Indian specimens, we think that they came through Korea as well. It may be via Sakhalin, Russia though. After invading Japan, MM5 expanded its distribution throughout Japan. And other haplotypes were derived from MM1. Conclusion: We sequenced 1585 base pair of 59 samples of the Japanese black-eyed population. 6 haplotypes detected from 59 individuals. 50 of them are the same haplotype. The 4 haplotypes differ by 1 base pair from MM1, while MM1 and MM5 differ by 3 base pairs. Japanese black-eyed group is divided into a group of MM1 and a group of MM5. Population genetic analysis showed that the Japanese black-eyed population is a population expansion model. Genetic diversity is lower than other birds of prey. The origin of the Japanese black-eyed considered 1. MM1's few individuals invaded Japan from the continent of East Asia. 2. MM1 spread all over Japan. MM5 individuals invaded 2. 3. MM2, 3, 4, and 6 are derived from MM1. 4. Fitting to life with humans and increasing population, now in the process. 6. Since there are observations that the population is now decreasing, it is possible that the population is now at its peak and will continue to decrease, and the genetic diversity may not increase much. As with other raptor species, we need to keep an eye on it. Thank you for collecting the sample. Thank you very much. If you have any question, email to me. Now this study is in submitting to an international journal. Thank you for your being. Thank you very much.
All right, we thank Kazuya Nagai for the interesting presentation. So since he won't be here to take questions, kindly contact him through the contact that he actually showed in the last slide. All right, so for the benefit of time, we'll move on to the third presenter, Oluwadunsin Adekola, who will be presenting on the title, Extent and Symmetry of Tail Moult in Amur Falcons. Oluwadunsin, once you're ready. So it will be a recorded presentation as well, but I believe he will be here to take questions at the end.
Kola. I am a PhD student at the Fitzpatrick Institute of African Ornithology, University of Cape Town in South Africa. My research focuses on moult patterns and feather quality in raptors. And today, I will be telling you one of the most fascinating and important parts of avian life, which is moult. And for the purpose of this presentation, I will focus on the extent and symmetry of tail moult in Amur falcons. Moult is one of the 3 major events in the annual cycle of birds. The 2 other events include breeding and migration. These events are energy demanding, and therefore birds tend to avoid overlap between them. In other words, because moult takes so much energy, it tends to take place between other costly events. Birds tend not to breed or migrate during heavy moult. However, unlike breeding and migration, all birds must molt because feathers are not permanent structures, which means that not all birds breed and not all birds migrate, but all birds need to replace their feathers regularly, and this include young birds, passerines, flightless birds, and birds of prey. Molt is beyond the replacement of feathers. It is an evolutionary and dynamic process, which is essential for various reasons, such as the replacement of old and worn feathers. You can see a typical wing feathers with a mixture of old and new feathers. The old and worn feathers would be dropped and replaced with new feathers, and this process is called molting. When the old feathers are dropped, gaps are created for new feathers to be developed, as seen in these pictures. These gaps affect flying and foraging efficiency negatively. So therefore, birds tend to minimize gap in the wings or in the tail when molting. Molt is also essential to change colors for territorial or sexual display. And a classic example is the male red foodie in non-breeding plumage. This is an example of a male red foodie in non-breeding plumage, and this is it during molt, and also this is it when in breeding plumage. Another example is the male Sopalfarian wren, this is the image during non-breeding season, and this is the picture during molt, and then this is the picture during breeding. The study of molt has important consequences for understanding bird biology and conservation, and research has shown various conservation implications of molt. And for example, the understanding of molt strategies provides insight into the plasticity and phylogenetic conservation of feather replacement, which can influence the species distribution. Molt patterns might be used as an index of condition linking to conservation. Molt is often used to determine birds' age, which is essential for understanding their population dynamics. And molt studies has been used to assess territory quality and as a novel tool to study population ecology. Also, molt extent and symmetry have been proposed as a predictor of fitness in birds. And finally, molt extent has been used to predict global warming and climate change. It is quite interesting to note that the time of molt, the location of molt, and the extent of molt tend to change more often in different species. In other words, they are very plastic. However, molt sequence is more fixed, except for wave molt. And most bird species undergo sequential molt, whereby feather replacement starts from the innermost primary and molts outward. However, in falcons, the sequence of molt is quite uncommon, proceeding bidirectionally in two molt series at both primaries and secondaries. Adult falcons typically molt their flight feathers in one molt cycle, that is within 1 year, but the timing and extent of juvenile flight feather molts varies considerably among species. For example, the sooty falcon, which is a long-distance migrant, replace all their flight feathers in their first winter. However, juvenile of other long-distance falcons do not molt their flight feathers on their wintering ground. The Eurasian ubes and early Hanover falcons, for example, undergo a similar migration to the sooty falcons, yet they only molt in their second summer. Other migratory species like the lesser kestrel and also the red-footed falcon only molt a variable number of tail feathers on their wintering ground, while retaining all juvenile primaries and secondaries. All this show the complexities and variations of molt patterns across different bird species. Our study species, the Amur falcons, undergo one of the most extreme migrations of any raptor. They travel over 14,500 kilometers and they breed in Eastern Asia and winters in Southern Africa. In March 2019, two hailstorms occurred in KwaZulu-Natal, South Africa, that affected almost 4,000 Amur falcons just before migrating back to their Asian breeding sites. One of the hailstorms occurred at Moore River on the 9th of March and another at Newcastle on the 21st of March in the year 2019. With the help of volunteers, more than 1,000 birds were rescued, and we eventually got reports that some returned to their Asian breeding ground. We scored flight feather molts from almost 2,000 adults and juvenile Amur falcons killed during the hailstorm in March 2019. We recorded more males than females across events and age groups, except in juveniles during the first event, where we recorded more females than male armor falcons. Male biased adult sex ratio is common across many bird species, particularly in threatened species. The number of tail feathers replaced in adults was bimodal, with no birds having replaced 6 or 7 tail feathers. We termed those that had molted 0 to 5 feathers as late tail molters, and those that had replaced 8 to 12 feathers as early tail molters. Surprisingly, we found that adults killed in the first event had molted more tail feathers than those killed 12 days later. We think that the bimodality of adult tail molt is traceable to the late molters being second-year birds, and their population could be higher in the second event if some had already departed KwaZulu-Natal by the 28th of March. Juvenile male armor falcons showed increasing progression of body moult into adult male plumage from stage 1 to stage 4. The bird in stage 4 is the oldest juvenile male molting into adult male plumage. And as you can see, as males get older, the paler their belly feathers. We also showed a positive relationship between extent of tail molt and body molt in juvenile males. In other words, as body molt increases, extent of tail molt also increases. There was strong selection for symmetry across tail feathers. Tail molt was much symmetrical in adults than juvenile. The central tail feathers, which is the T1, had lowest symmetry in adults and shared lowest symmetry with T2 in juvenile armor falcons. The greater asymmetry among juvenile tail molt might indicate selection for birds with greater symmetry. For feather replacement, molt typically started with the central tail feathers in both adults and juvenile. However, adults were more likely to have replaced their other tail feathers than juveniles. We found variation in the sequence of tail molt, with most birds either molting outward or inward. Outward molt is when molt starts at the center and proceeds outwards, while inward molt is when molt starts from the outer tail feathers and proceeds inwards. In adults, we recorded 67% molting outwards and 24% molting inwards. But in juvenile, 97% were molting outward and only 1% were molting inwards. Surprisingly, those adult molting outward mostly consist of late molters, and most of those molting inwards were early molters that were close to completing tail molt in mid-March. This variation in molt sequence may suggest molt plasticity and may be an adaptive response to the problem of optimal timing of molt of differing feathers within the same feather tracts. The extent of tail molt correlated with body condition in both juvenile and adults, which means that birds with better body condition tended to molt more tail feathers, suggesting that molt patterns might be used as an indicator of fitness in falcons. On a final note, despite the reverse sexual dimorphism that has been reported in birds of prey, that is, females larger than males, and other morphometric measurements such as bill, head, wing, and tail length longer in females than males, we found that male armor falcons have longer tarsus length than females in both juveniles and adults. This unexpected finding may relate to a more area hunting patterns in males, possibly related to provisioning during the breeding period. In conclusion, moult is an important process in avian annual cycle. Moult patterns might be used as an indicator of fitness in falcons. Further study is needed to investigate the causes and consequences of the various tailed moult strategies found in armor falcons. This research has already been published in the Journal of Ornithology. I will advise that you check it out for more information. I would love to acknowledge the support of the following organizations: the Fitzpatrick Institute of African Ornithology, the Department of Biological Sciences, and the University of Cape Town for funding and logistics, the National Research Foundation for funding, Agnes Bayern Foundation for funding, the staff of FRIMI KwaZulu-Natal Wildlife Rehabilitation Center for assistance in making the falcon carcasses available, and the Durban Natural Science Museum in South Africa for help and logistics. Thank you so much for your attention, and I await your questions.
Thank you, Oluwadunsin, for the very interesting sharing. Do you have any questions, please? If you have questions, kindly unmute yourself to ask your questions, or you can just type it out in the chat box, and I'll try to read it out. Oluwadunsin is here in person to actually answer our questions. While waiting, I have actually a simple question to ask. So correct me if I'm wrong, from your presentation earlier, I understand that these patterns were actually detected from the falcon carcasses that you have obtained for your samples. Have you actually compared the patterns observed here with patterns of the moult patterns in live falcons? Oluwadunsin.
Thank you so much for your question. I hope you can hear me.
Yes.
Yes, thank you. Yes, that is the next plan now. I already had a relationship with somebody, so kind of collaborating. She collected moult data from live armor falcons, and the next thing we're planning to do now is to compare not just differences between dead and live birds, but we want to also compare for moult patterns between birds in South Africa and the ones that are breeding over there as the Eastern Asia. So we're trying to see exactly what is happening between, if there are some birds that are molting on their breeding site, as well. And we're trying to do this comparison, and hopefully in the nearest future we'll come up with a result from there.
Great. Thank you very much.
Thank you so much.
You also mentioned that the extent of molting could possibly relate to the fitness of the species or of the individual birds. But what about the direction of the moult itself? You mentioned patterns of molting outwards and molting inwards. Are there any kinds of evolutionary significances or context that you can put this pattern in?
Yeah, that's a very beautiful question, and thank you so much for that question. One of the things I noted, the conclusion is that research still needs to be done to know the consequences of the variation we'll see in the moult strategies in armor falcon. Absolutely, we can use direction of moult to talk about the phylogenetic and plasticity in birds. There was this study by Peter Pearl that actually talked about the evolutionary replication of moult between falcons and parrots. And then from there, from their pattern of moult, you can see this evolutionary connection with them. So birds that are actually from the same lineage, they tend to molt in a particular sequence, and compared to birds that are far apart evolutionary wise. Yes, absolutely, I agree with you on that. And then since we can use mood patterns to as an index of condition and to predict fitness. I strongly believe as well that we can, but though we didn't do that in the armor falcon because we're not comparing with any other species, but we can actually use that as well, both the sequence and the extent of modes, even timing of modes. One of the, I'm sorry to, maybe I'm taking your time, but one more thing I would love to add is one of the studies I did on other species entirely. We found that the timing of mode is more earlier in birds that are in good body condition, and the molt faster, so they replace their feathers faster if they're in good body condition to birds in location that they have high competition for food, they're open to more predations and all of that. So it tells on their body mode. So we can use mode patterns as a non-invasive techniques to know, to predict habitat quality and also to determine the health of a species.
Very interesting, very interesting. Thank you very much. And all the best to your next endeavor.
Yeah,
all right.
Thanks.
So if there are no further questions, maybe we can move on to the next presenter. Yeah. So next we have Andreas Bandang Hardian, who would be presenting on the title Blood Cells Morphometry and Descriptive Morphology of Captive Changeable Hawk Eagle at Wildlife Rescue Center, Jogja. Andreas, when you're ready, kindly proceed.
Thank you very much, Dr. William, for the presentation of that person. So can you see my screen, and is my voice clear?
Yes, I can see your screen and also your cursor, but your voice is, it feels interrupted. Could you maybe, adjust a little bit?
How about now? Is it okay?
There's a bit of interference going on.
So yeah, just in that go back to so maybe I can be using all of these subjects.
Okay, kindly proceed. Thank you.
And yes, good afternoon, good morning, and good evening for those are in the different time zones. And my name is Andreas Bandang Hardian. Here I'm working on behalf of the Wildlife Rescue Center Jogja. And this is the fifth project of Yogyakarta Nature Conservation Foundation. We are located in Yogyakarta, Sumber Java, Indonesia. And it just moved from the other room, so just a bit awkward here. But here I would like to present our part of this research. So last 2 years, we worked with Church Abu Haf eagles and some conservation medicine of it. And this is part of our purpose to explore more about the hematology of this eagle, because we found that the Church Abu Haf eagle is quite abundant in Indonesia in every rescue center. And we thought that some of the health problems occurs out there, and no one can decide from the hematology, because the hematology reference interval is yet not exist in Indonesia. So we try to initiate it. So our title will be the blood cell morphometry and descriptive morphology of captive Abu Haf eagles. And a bit about our center, we'd like to introduce you about our place. So this is our center, and this is the FIT project, like I told you before. It's from the Yogyakarta Nature Conservation Foundation, and we are located in Yogyakarta and covering the working region in Central Java. We are doing rescue, rehabilitation activity, and also daily attempting to be part of research. And yes, we intend to be a research center in the future, and now we are affiliating to several universities in Indonesia. And you may check our website and see what we are doing and what is going on there at the moment if you are interested in working with us. And this is the background and the methodology of our study, providing qualitative and quantitative hematologic reference of neglected captive wild animals, because in Indonesia we know that we have some charismatic animals that get more concern from the researchers. And now we talk about the diplomatic one. And yes, we are in the concern of these species. Many species of our endemic animals are neglected because of the charismatic always get more focus from the other researchers. And in terms of this, we dedicate ourselves to learn about the diplomatic one. And we know that Church Apple House eagles are abundantly kept in the sanctuary with no hematologic reference is ever reported. So that for the diagnosis according to the hematologic value is quite inaccurate at the moment because we have no reference interval. And we would like to provide the key of morphological identification because this is the part of the hematologic test. We need to know about the morphology and also the identification of each blood cells. Then we collected and prepared the peripheral blood smears with a tenfold dilated KIMSA stain. So it's quite simple and also very classic way to create the blood smears from 8 changeable half eagles in wildlife rescue center Jogja, following the standard manner. And all slides were observed and captured under the camera with microscope. So we use the Optilab, that's the initial product of the microscopic camera for histological matters. And the evaluation of the morphology and morphometry aspect were conducted using image separation 1.52A. And as the changeable height equals are naturally present as dark and bright moths in nature. So we also group some data according to this dimorphism or the variation of the feather scales, and we will compare them statistically among the groups. And this is the result. This is the result of our measurement of the cell morphometry. So you can see that. So this column explains about the mean and also the deviation standard of the cell length, width, length, and the close length and close width of the erythrocyte. So the erythrocyte of the dark moth and the bright moth of the birds is different, but when we calculated it statistically, it showed that the p-value is less than 0.5, so that is quite significant. And then the other part is the white blood cell. We have some of white blood cell here. We know that the white blood cell is quite amoeboid, or the irregularity is quite common in white blood cells, so that we just working out for the significant differences. And then the digital cell morphometry is done in 240 erythrocytes, so it's quite many, more than 100 erythrocytes, excluding the basophils, because we only found about 5 cells of basophils here, but it's quite difficult to discriminate from the others. And more than 200 thrombocytes as well, we have research in this study. And this is the pictorial guide. So I would like to be straightforward for this. And you can see that actually all of the morphological characters quite common for avian, but maybe you would see and appreciate some specific feature for the eagle. Yeah? So the erythrocytes were oval with a clear cytoplasm, you can see here. And the nuclei were stained purple to magenta with a distinct dark blue chromatin, and the circulating RBC-like cells were found occasionally, which were later identified as the immature erythrocyte. So on this one, so actually some of the mechanism in our body will fill the gap of the mature erythrocyte scarcity with the immature one. So you can see the immature erythrocyte in the peripheral blood cell like this, and it's a bit basophilic in the cytoplasm, and the nucleus is also a bit larger than the mature one. But when you see this one is the thrombocyte of the part. So that's a bit different from the erythrocyte. So they are same as oval shape, but this one is quite smaller, and the cytoplasm is not quite distinct here. And then in bird, we know that they don't have any neutrophils like in mammals or in the higher taxa of the animals. We call it the heterophils. So in this one is the group of heterophiles. You can see also here that the heterophiles show no distinct cytoplasmic granules actually, but they indeed have the granules here, but the granules is not quite pale, so that it's not like the distinct as the eosinophils here. And then you see that the eosinophil has a very prominent and distinct granules, the reddish granules that will be in the very specific pattern of each eosinophils. But eosinophil sometimes occur in variation of size here. So there's small one and also the big one. And sometimes they also show it in the peloculated nuclei like this. And then the last one is the basophils, the bright magenta stained small irregular spherical shape with the indistinct round face and occasional cytoplasmic granules protruding to the cell membrane. So you can see that we only have 4 here for basophils because it's quite difficult for us to find the good picture of it, because sometimes some of them just burst out when we made the blood smear. And this is quite distinctive rather than the other cells because it's quite smaller. And the most specific feature of this cell is this. Although this is a leukocyte, but the nuclei is unknowable. So like this one. This one is hypersegmented or multilobulated, but this one is oval. In mammals or in the Hyracoxa, we have the basophil, which shows the lobulated or multisegmented nuclei. But here in bird, especially in this eagle, is not. And then we have also the lymphocyte groups here. So lymphocytes is usually producing the antibodies, and this one occurs in many size of a shape, from the smallest one to the biggest one, because they usually sensitize by the antigen to create the antibodies inside the cytoplasm. So the size may be varied. And then the monocyte occurred as large cell with dark purple ovate around peloculated nuclei. So in this one is the classic presentation of a monocyte, but we also have the variation like this. So the size is also vary, and also you can see some of them have the love heart shape here, and they can be vary with this variation as well. And then the thrombocyte, the last one, or we call it as the platelets, were the mostly found in the aggregate consisting of several cells. So they can pop as this one, like this aggregation of the platelets, but they also occur as the individual one or the solitary cells. And this is just a pictorial guide. So actually, this should be appreciated before we did the hematologic test. So after we are able to recognize every cells here, we will be better to do the hematologic test, because every differential leukocyte or every hematologic count for RBC or WBC will depend on our skill to discriminate each other of this cell. And the conclusion is overall the blood cells morphological apparatus of the gingival hatical are visually identical to other avian species, but it is morphometrically diverse. And several studies reported that the morphometry of avian erythrocyte might differ among birds, but due to the variation of activity, daily diet, genetic, and environmental conditions. And the statistical comparison of erythrocyte and leukocyte morphometry between the dark and bright mobs of the genes of the showed a significant difference. So I think we need to ensure about this fact, because this is actually the same species, but maybe there are some risk factor or maybe in the environmental factor that affect the size of these erythrocytes. And here we roughly categorize the changeable health equal lymphocyte size based on the visual inspection of the histogram and strip chart into the three groups: the small one, the medium one, and the large one. So you can see in the histogram here actually. So I think I missed this one. This is the histogram of our categorization of lymphocyte size. So they are the smallest one and the medium size and also the larger one. According to this histogram, we can assume that this group is not sensitized one. So we call it as a naive lymphocyte. And this one is the strongly sensitized one. So in this one and also this one have met the antigen and produce the antibody inside the cytoplasm. And then I think that's all of our presentation, and we'd like to thanks to some parties that support our research and study. First, for the Ministry of Environment and Forestry of Indonesia for permitting us performing this study, and our counterparts from the university, the Faculty of Veterinary Medicine, Universitas Brawijaya, and Universitas Gadjah Mada for assisting us and lend us the camera and also some reagents for free for this research. And if you are worrying about us, you may visit our social medias as presented in the screen. And if you have some intention to have research collaborating with us, don't be hesitant to contact us in the future. And that's all of our presentation. Thank you very much, and I will give back the time to Dr. Will.
Thank you very much, Andreas, for the very interesting insight on the blood profile of the changeable hawk eagle. So any questions for Andreas on the study? You can turn on your mic to actually ask questions, or you can just type it out in the chat and I'll read it out. While waiting, maybe I can ask a simple question also. I see a very diverse blood profile, and you actually summarize it. In your conclusion, you also mentioned that in this species that you guys found, you see a very diverse kind of profile in terms of the different kinds of blood cells, even more than some other bird species. And this is usually due to diets and activities and genetics and all sorts of reasons. But since the birds are currently in captive, is there a reason why they are so diverse in terms of their blood profiles?
Okay, that's good question. Yeah, that's still in our observation actually. If the captive condition will affect the variation of the morphologic characteristic of this bird blood. But in fact that some of the basic condition of blood shape will be dependent on the genetic. So we know that the captive condition will not affect strongly the blood cells, but more often about the diet and nutrition. So that will be sufficient nutrition or not, or maybe some toxic material from the food that maybe alter the shape of the blood, or maybe in our center we have some mosquitoes there that maybe bring some blood parasite or something that maybe alter also the shape of the blood cell. But yes, that they still further research for this, and this is just initiative of us to create this pictorial guide, so that it will be make some easy identification of hematologic tests.
Mm-hmm. So are the birds from various places within Indonesia, or is it just around Jogja that you have in your center?
Actually, we have the regional bird from Java only at the moment. But last year, we also have the cassowary from Papua, but most of them actually from the illegal trading. So we just rescue them, they transit in our center, and we send back them to the original place of the epidemic.
I see, I see. All right, thank you very much, Andreas, for the very interesting sharing. So far, we still do not have questions, but if you have further questions, I mean, to the participants, feel free to contact Andreas to ask your questions. All right, I've been made to understand that our final presenter, so thank you, Andreas. I've been made to understand that our final presenter, Abdul Rahman Hanif, is not able to attend today, I believe. So our concurrent session 8 will end here. All right. So we just heard the last presentation from Andreas. So I would like to thank all presenters for your very interesting sharing. I believe every one of us here have benefited greatly in terms of understanding the genetics, whether it's genetic variation, among populations or within a small population or even within individuals, all right, of the different species of falcons that have been shared. And also to participants to actually attend the concurrent session today. So since we have ended here, kindly proceed to either the main room, or you can still proceed to room number 1, okay, to listen to the concurrent session 7 over there. All right. Thank you very much, everyone, for attending.